Ladies and gents, this is the moment you've been waiting for. Another biology with Miss Judd. We just learned about those awesome discoveries of Gregor Mendel. If you do not think he is fascinating, then rewatch that video to make sure that you are fully team Gregor Mendel, because Miss Judd is. Today, we're going to take his patterns and what he found out and really kind of dive into and see the modern applications. What this means is that we will be learning some content, but really we're going to be learning a lot of skills. We're going to be learning how do we now use those patterns to make predictions. So buckle up. It's going to be a lot of information. You've got this. I'm so proud of you. Let's send it up to Miss Judd in the corner. Hi, you guys. Miss Judd in the corner here. Let's dive into Unit 5, Lesson 2. We are going to start off by just reviewing what Mendel found out for us. So he came up with these 3 rules. The law of segregation, where a parent who has 2 alleles for a gene can only pass on 1 allele or the other to each offspring. And then that leads to the law of independent assortment. 2 or more traits are inherently separate from each other. They don't always occur together. And then 3, the law of dominance. 1 dominant allele will take charge or take over the recessive allele and mask it, right? So we have these 3 laws that Mendel taught us. And then we can boil this down, and what we're going to dive into today is kind of how these genes and alleles work together. Remember that a gene is a segment of DNA that codes for a very specific trait. For example, a trait, we looked at those. Can you roll your tongue? When you cross your hands, is your right thumb on top or your left thumb on top? The alleles are the different forms of a gene. So organisms inherit 2 alleles for each trait, 1 from each parent. So you get 1 allele from mom, 1 allele from dad. And these alleles can either be dominant or recessive. They can either take over or get hidden. Remember that Mendel, in order to find out these things, looked at P's. We got a bunch of P's here. So let's take this idea that alleles for traits are either dominant or recessive. The dominant allele is expressed when it is present. So we represent it with a capital letter. So if we look over here, capital B is that dominant allele. And it covers up the recessive allele for a given trait. The recessive allele is a lowercase letter. It is masked by the dominant trait if that dominant allele is present. So the only way to express that recessive trait is if there are 2 copies of the recessive allele, 2 lowercase letters. Because as soon as you put one of these capital letters in front, you're going to see that dominant trait. Let's kind of walk through this with this example in the corner. We have 2 alleles, brown and blue, for the trait or the gene eye color. Brown is dominant over blue. So we have 2 alleles inherited, 2 capital B's, so that offspring is going to have brown eyes. In this case, we have 2 letters. Mom gave a lowercase b, but that gets hidden by the capital B inherited from dad. So this offspring is going to have that brown eye, that dominant trait. In this case, we get 2 lowercase letters, 1 B from mom, 1 B from dad. So this person will express that blue eye color, the recessive trait. What we are kind of diving into is genotype versus phenotype. So that physical expression, whether or not you have blue or brown eyes, that is the phenotype. When you think P for picture or physical description, phenotype. So an example here would be either being tall or short. That's your phenotype. That's physically what you look like. Now, how we determine this phenotype is by looking at the genotype, the actual specific alleles an individual has for a gene. These are the letters. So big T, big T, big T, little t, little t, little t. Based on that genotype, the genes, what are they going to look like, or phenotype? We have 2 types of genotypes. We have homozygous. Homozygous, homo meaning the same, means that they have the same allele from both parents. So they could be homozygous dominant, two capital A's, homozygous recessive, two lowercase a's. In homozygous, they have the same letters because they're inheriting the same allele from both parents. This is also called purebred. Heterozygous is when an organism has a dominant and a recessive allele, big A, little a. Individuals are heterozygous for a given trait are called carriers. Think back to our sickle cell conversation. Carriers don't show the trait because that dominant is in front, but they carry it behind them. This is also called hybrid because it's a mix. So hetero, different, you have two different alleles, two different letters. Now, in genetics and in heredity, we are actually able to say how likely a trait will get passed on to your offspring. And this uses this idea of probability. Probability is the ability to measure how likely something will happen. So we can take two parents and say, of your possible offspring, so we have two parents here, and of all the offspring, we can say that there's a 25% chance that your offspring is going to be white instead of purple. And in order to get this probability, we say the number of one kind possible outcome. So the number, this is our offspring. How many are white? 1. Out of my 4 total outcomes. So 1 out of these 4 is a 25% chance. So we are taking this idea of how likely something will happen when we cross two parents together. And we use a tool to get to this probability. The tool that we use is called a Punnett square. This makes predictions about the outcomes of a genetic cross. So we use the probability, the likelihood an event will occur, to predict possible genotypes and phenotype combinations of offspring. Now, Ms. Judd has created a great document called Pardon the Punnett. And I think the best person to walk through Pardon the Punnett would be Ghost Ms. Judd. Pardon the Punnett is just some guidelines on how to do a Punnett square. So the first thing you want to do is state what the symbols or the letters mean. Big T means tall, little t means short. Then you write the genotypes of the parent organisms. So I'm crossing together big T, little t with little t, little t. Then you draw out the Punnett square. The Punnett square, you write the parent gametes on the side and then the top. So big T, little t, I separate it out. Big T goes on the side and then on the other side. Little t, again, one, and then the other square. So that we could complete the square by combining the mother and father gametes. So big T goes with little t goes with little t. Big T goes across, little t comes down. Little t goes across, little t comes down. Once our Punnett square is completed, we can figure out the results of the genotypes and phenotypes. So, as I have 0 big T, I have 2 big T, and I have 2 little t. So I have a 50% chance of it being tall and a 50% chance of it being short. All right, let's practice this out. We have the situation, H is for Harry, so I'm going to write that down. Big H equals Harry. Little h means no hair, so I'm going to write that down. Step 1, write what the letters mean. And then my genotypes of the parents are already written for me. I just have to split them. Big H splits, and then my mother genotype, big H, little h, I can split that. I'm going to use a different color. Big H goes on one square, little h goes on the other. And then I combine. So one from mom, and then a big H from dad. And then a big H from mom goes down, a little h from dad goes across. And then I move over, a big H from dad goes across, a little h from mom pops down, and then a little h from mom, and a little h from dad. Now that my square is complete, I can write the probability. So the first thing I'm going to do is how many big H's do I have? 1. How many heterozygous, big H, little h? 2. How many homozygous recessive? 1. Now the probability part. 1 out of my total 4 is 25%. 2 out of my total 4 is 50%. 1 out of my total 4 is 25%. All right, so we've just gone over a couple of information, and it's a lot. So let's take a look and practice it. In your guided notes, your practice looks like this. It says part 1, and then it lists it out. When we are trying to determine a phenotype based on a genotype, we are writing the physical description based on the genes we are given. So we are told brown hair is dominant to blonde hair, which means if I see the dominant allele, this capital B, my phenotype is going to be brown. Because I have this capital B, that dominant allele, my phenotype for this genotype is going to be brown. Because in this next one, I have 2 lowercase b's, 2 of those recessive alleles, it's going to be that recessive trait, which is blonde. Now we can flip it. We can say, list the genotype next to the given phenotype. And we're told the pattern, purple flowers are dominant to white. So if I see a purple flower, it absolutely has to have that dominant allele of a big P. But there are 2 ways that could be. It could be homozygous dominant, big P, or heterozygous, big P, little p. To be white, to show that recessive trait, I need both recessive alleles, little p. Now, when we are writing this and handwriting it like we are supposed to be in our guided notes, when we write the letter P or the letter S, the capital letter and the lowercase letter look so similar. So if you draw a line over the top of the lowercase version, that's a good way to say, like, I'm squishing this down. This is the lowercase p. So to differentiate when you're handwriting, draw those lines on top of the 2 lowercase p's. All right, so let's keep practicing. I know it's not in the packet, but the only way we get good at this is if we practice. Let's list the phenotype next to the genotype. Freckles are dominant to no freckles. So if freckles my dominant trait, when I see that dominant allele, I know that I'm going to see freckles. So big F, I'm going to see freckles. Big F, I've got that dominant allele, I'm going to see freckles. Little f, nothing dominant, so I'm going to see that recessive allele, which is no freckles. Are we getting the hang of it? I hope so. As always, if you need extra help, reach out to Miss Judd. She's here to help you. She has examples out the wazoo. All right, so back to our notes. We are going to practice Punnett squares. All right, we have our Punnett square guidelines. We are going to practice this out. As Miss Judd in the corner said, the only way we get better at this is if we practice and practice and practice. So once you have completed your practice, if you're still confused, please reach out, send an email, join an office hour so you can get more help. We will be practicing this some more. Make sure that you completely understand this. We will be coming into next class, doing more practice, and then our lesson check, so be prepared for that. I am so proud, as always, of all the work you're putting into this. Have a great day, and make good choices.